Hey everybody, I thought we could take a minute to review some of the material new to this week. I had you watch a couple of videos about DNA to review some of the structural differences or maybe some things you remember from freshman year. So this is all part of our heredity unit where we're going to be looking at how information is passed, how it's stored, and then how it eventually is going to turn into things we know as genetics, how it's going to determine the physical features amongst us. So we've already really covered mitosis and meiosis from before. So this week, and today especially, we're going to focus on DNA. So just to mention a couple things about DNA, DNA is kind of the set of instructions, and it's what we, pretty much identify as our genetic code and all of our genes, but DNA in and of itself doesn't really do anything. The RNA is going to be the material that actually turns your genes into something usable for the cell, so something tangible. RNA is what creates all the proteins that we've been talking about, proteins that control things in the cell cycle, proteins that control physical features about your cell. So DNA is the code that gets passed from one generation to the next, but RNA is something that's generated from the DNA so that you can turn into the organism that you are. Okay, so a little bit of a history about DNA. I thought I'd start with prokaryotes. I hope by now that you remember that prokaryote is another word for bacteria. And since we've been talking about evolution and how bacteria were probably the first organisms on Earth, the oldest fossils of bacteria date somewhere around 3.6 billion years old. The way that they have had their genetic information present hasn't probably changed much in that time. So this is just one particular example of one shape that a bacteria can have. This is called a bacillus bacterium. Single-celled, so unicellular, no actual nucleus, but there is DNA. So this is their version of a DNA. It's actually one circular chromosome, so it's a little bit different than eukaryotic chromosomes. There are some instances in which you will find that some bacteria can actually have two circular chromosomes, and in some really odd instances, 3 circular chromosomes as well. The numbers that you see here, 2.1 MB, the B stands for base pairs. So base pairs are the numbers of bases that you would find. So I think you remember that like A and T is a base pair and G and C is a base pair as well. So we're talking 2.1 million base pairs. So they're still really long, nowhere near as long as a eukaryotic cell. But nonetheless kind of important. So the way they replicate is different. We know they don't do mitosis because they don't have a nucleus. They will simply just copy this chromosome and then they will split in two in a process called binary fission. So binary fission is the method in which bacteria use to reproduce. This little guy here called a plasmid, we're going to talk about him a little bit later. Plasmids are kind of interesting. It's extra genes that are found in all different kinds of cells on Earth. Okay, so now on to eukaryotes. All right, so eukaryotes include plants and animals. They also include bacteria. No, they don't include bacteria. They include fungus and protists. So most of them are multicellular like us, but some of them can be unicellular like some of the fungus and some of the protists. But the big classic case here is that our DNA is found inside of a well-defined nucleus, and that nucleus has a membrane around it. So if you look at the picture here at the bottom, this purple, this is known as the nuclear membrane. Some textbooks and some authors will call it a nuclear envelope, or envelope, if you will. Envelope, couldn't spell that one. And also what you'll notice is that there's holes. There's pores in the nucleus, just like there would be pores in your skin. And this is important because there is stuff that has to get out of the nucleus every once in a while. So here's that chromatin we talked about. So clearly this is a cell that is in interphase. It legit looks like spaghetti. when DNA coils up, this is kind of showing you how it does that. So here's your double helix, which I think you're familiar with. The double helix will wrap around little proteins called histones, and then those proteins wrap around themselves and they form what's called a nucleosome. So what's happening is we're tightening, we're coiling, we're shortening, and we're thickening all this chromatin and DNA together, so then it becomes very solid and visible. So pretty much it would look like this, all condensed together. There's that centromere, which is that protein glue that kind of holds both sides together. And a little bit later, we're going to focus a lot on locations of genes, and we often identify locations based on what arm they're on. So up at the top here you have, I can't believe P and Q are back, but they are. So P represents the short arm of a chromosome, and Q represents the long arm. So when scientists identify the location of a gene, they will identify it by numerically by P and Q to kind of particularly identify where they are in a chromosome, or where they are on a chromosome. Okay, I just want to add in a few more historical things about DNA. So DNA itself wasn't really finalized until 1953, but before that there was a lot of experimentation going on to try to figure out what it is inside of a cell that transmits information. So they had this idea of something called a transforming principle. So Griffith was the first one to try to work on that particular experiment in 1928. And there's all kinds of detailed information about what he did, but he basically modified and manipulated various forms of materials, not knowing, of course, that it was DNA yet, but knowing that there must be something in there that is transforming a cell into what it was. So he did work with some mice and so on. Then there was another group called Avery, McLeod, McCarty in 1944, and they were the ones that discovered that it was in fact something called a nucleic acid. Now, of course, we call it DNA, but it was a nucleic acid, meaning it was found in the nucleus, and it was in fact not a protein. So before, it was really thought that the transforming stuff that we pass from one cell to another is protein, but these two experiments confirmed that probably was not the case. And then another experiment came along in 1952, or the Hershey-Chase experiment, which really solidified this idea. Hershey and Chase used viruses in order to support this hypothesis. So once again, they showed more evidence to support that this genetic material was not in fact a protein. What they did was, this is actually a picture of a virus. This is not what all viruses look like. This is called a phage, and particularly it's called a bacteriophage. It looks like a spaceship, but it's a virus that infects bacteria. So this would be your bacterial cell. So believe it or not, even bacteria get viruses too. So they radioactively added material to the protein coat around the virus in one experiment, and then they radioactively labeled this other stuff inside what we now know, of course, is DNA or RNA, but this genetic material inside. Then they let the phages do their thing and try to infect the cells, and then they tried to figure out, well, did the cells pick up the radioactivity or not? And only in the case of the fact where the DNA was radioactively labeled did the cell retain that radioactivity. In the other ones, the protein code is not transferred into the cell, so therefore we know that that is not part of the genetic material. Okay, so that was a little bit of a history surrounding it. Now to the more familiar things you're more likely versed in, and that's Watson and Crick. So Watson and Crick finalized the model of DNA in 1953. So DNA, of course, I hope you know by now, stands for deoxyribonucleic, which is all one word, acid. So it's a good thing that they shortened it up to DNA. It's an acid, but it's not the same type of acid that you were thinking about. It's a pretty neutral acid in and of itself. So really what they did was a lot of trial and error trying to build this model, and they built it the same way you would in a chemistry classroom with ball balls and sticks and tried to match up electron bonding and make sure all the cells are kind of doing what they're doing. And they had a lot of wrong models too. One of the first models they had was they had all the bases kind of hanging out on the outside, so like what we now know as A and T and G and C, well, that would be wrong anyway, were on the outside, and that wouldn't work because we have all these available electrons that would not be satisfied. So lots of trial and error. Eventually they got the information that they needed to finish the model, and they got that information from two additional scientists, Rosalind Franklin, which is this lady here, and then Maurice Wilkins. I did not include his picture because he looks like a typical old white guy. Rosalind Franklin's kind of famous now because of this right here. So this, she actually wasn't even working on DNA. She was working with a technique called X-ray crystallography, where she was taking X-rays of really, really tiny materials to try to figure out their structure. So you can read about the crystallography. It's kind of fascinating, but not really necessary for the course. But one of the things she did is she took some of that transforming material inside the nucleus of the cell and diffracted it and came up with this photograph. And this is very famously called Photo 51. And Photo 51 is very telling. It tells us a lot about the shape of the double helix. It is a little bit unknown as to how this photograph got into the hands of Watson and Crick. Some might say that it was given to them by Wilkins. Maybe Rosalind Franklin cared, maybe she didn't. No one actually knows for sure. But as soon as they saw this photograph, this would be like as if you were looking down the center of the double helix, the winding staircase right down the middle, and these crisscross patterns you see are the twisting. So this is what allowed Watson and Crick to say not only is it a helix, but it is in fact a double helix. And then you can see the base pairing objectives there as well. So that was a very, very important discovery. Eventually won the Nobel Prize. Unfortunately, Rosalind Franklin did not win the Nobel Prize because it was awarded after her death. She died of cancer. Some may attribute that to her excessive use of X-rays. And now, of course, we know a lot more about X-rays and radiation, but at the time, not much was known. So she is cited in the paper as a contributor, but we don't know her as well. But it was really this picture that allowed the whole model to be completed. Okay, last few things here, getting into the structure of DNA. So DNA is a really long molecule. We were looking at the bacteria earlier where they have billions of nucleotide pairs. The same goes for us. So DNA is made up of repeating units. It's a polymer, okay, so it's something that repeats over and over again, made of a nucleotide. So down here is just one blown up picture of a single nucleotide. The nucleotide has three parts to it. A pentose sugar, PS, things that end in OSE are going to be sugars. This sugar is called deoxyribose. Okay. The sugar is bonded to a phosphate group, which is a phosphorus surrounded by oxygen. Notice that a few of those are negatively charged. That's a really big deal. And then lastly is a nitrogenous base. And there are 4 of those bases, A, T, G, and C. And there's actually a 5th one called U, but that's only found in RNA. Okay, so when you're putting it all together, you've got your pentose sugar bonded to one of your bases, and then on the other side will be the phosphate. So when you have this phosphate sugar kind of thing repeating, like there's no variation. These are the same 2 molecules over and over and over again. This is called the sugar phosphate backbone. Okay, that's the sugar phosphate backbone. Just like your backbone would be bone, cartilage, bone, cartilage, bone, cartilage. Same idea. It doesn't change. What actually is unique is down the middle. So the bases give you your uniqueness. So ideally, as long as you don't have an identical twin, no one else should have the same sequence of bases that you do. We do have a lot of the same, like 97% of my DNA is exactly the same as yours, but there are places in which variation takes place. What holds the 2 sides together is something called a hydrogen bond. So you can barely make out these little red lines between here. This is the hydrogen bond. It's really just an attractive covalent bond between the 2 sides. It's very weak. So they're easily broken, and that's important when it comes time to make copies of DNA, and then also when it comes time to make proteins from them. G and C have a triple bond, and A and T have a double bond. That's not really that important. Okay, the last thing I want to mention is this thing called 5' and 3'. So the two sides of the double helix actually move in opposite directions. So let me show you down on the next one here. This plays an important role in DNA replication. I can zoom in. Okay, here we go. All right, so barely you can make that out in red there, but the 5' and the 3' refer to which carbon in the carbon cycle here, in the carbon sequence, is the first one to start. So in this particular one, it's the number 5 carbon, and then you can kind of make out at the bottom here, this is the number 3 carbon. And on the other side of the molecule, it's reverse. So here's the 5', and then here's the number 3 carbon. So what does that mean? It basically means the two sides of the double helix are upside down, or what we like to refer to as anti-parallel. So the DNA double helix occurs bidirectionally, or what we call anti-parallel. One side's right side up, the other side's upside down. That has to do with this bond right here. The way the G's and the C's and the A's and the T's fit together, one of them has to flip over on its back in order to make that happen. When we copy DNA, which of course we know now is really important in terms of mitosis, we want to make sure that the copies are done correctly. Each strand is going to separate down the middle here. These hydrogen bonds will break. And then each strand will serve as a template for a new strand. So like a template is if you were doing a presentation and like you had to do PowerPoint or Google Slides, and you pick your background and your font, and then you just have to type in the words. That's the template. Like it's all basically set up. You just have to fill in the blanks a little bit, and that's how that works. So last couple things here. Oopsie. DNA replication. So here's probably the most important slide about replication for you. Looking at, make this a little bit bigger. Okay, so there's a few enzymes that I want to highlight. Remember, enzymes tend to end in the suffix ase. Okay, so you're going to see a lot of them. First one is helicase, super appropriately named. Helicase's job is to unwind the double helix, so it actually breaks the bonds between the base pairs. Okay, so that's the first thing that happens. Then you have something called DNA polymerase, poly meaning many, and its job is to just add in free-floating nucleotides where they fit. So like right here now you have a naked A, so this A has no friend, and therefore its electrons are not satisfied. So it's going to be colliding and searching for something that will satisfy it. So it will find a T, and a T will bond in there. So DNA polymerase adds and helps to piece that story together. The same thing is going on the other side of the naked double helix as well. So polymerase's job is to add the bases. We have another enzyme called ligase, and its job is to seal back all the bonds together, and pretty much how it does that is it adds some of the phosphates back to stabilize the molecule. And then we have a lot of proofreading that happens. So polymerase does a lot of proofreading as well. And what you want to make sure is that, you know, an A didn't accidentally bond with a C, because of course that would be bad. So you would have an enzyme that would come along and hopefully replace them. So that's how we try to fix most mutations before they even happen. Now, each double helix that you make consists of one original strand and then one newly formed strand. So here's part of the original, and then here's the newly made one. So what we call this is semi-conservative. So to conserve means to keep, and we're keeping half each time, so it's semi-conservative. So because DNA is like 3 billion base pairs long, I mean, the human DNA at least, this would take a really long time if you went straight down from the top. So the solution in eukaryotes is, well, let's start replication in a bunch of different places. So there's multiple origin sites. So it'll like open up here, it'll open up there, it'll open up there, and they'll continue to synthesize and copy in both directions until everything is kind of finished at the end there. So that's a way to save time. Otherwise, that S part of interphase would take a really, really, really long time to do so. Okay, I'm going to give a little bit more detail about how this works. I've got some videos I want you to watch because it's really hard to picture it in action unless you can actually see it. But we'll talk about this another day, the more details about how DNA is replicated. I just want to mention one final thing here. There is other place to find genetic material besides just DNA and RNA, and it's something called a plasmid. And we're going to work a lot with plasmids in the next unit because plasmids are really good for manipulating, and we can like add and subtract and manipulate genes. But plasmids are found everywhere. They're found in your DNA too. Sometimes it's not even human DNA. You actually have pieces of, bacteria that you have been infected with since you were a kid, or viruses that you picked up. You retain their plasmids, and then you can transfer those plasmids to new cells as well. They're really small, but they do contain some genetically unique information. Okay, so I think that's good for now. More to come later on DNA replication. See you soon.